Students, to start from where we left off, uh, we were studying about the characteristics of the cell. So let's look at what's there in a typical cell. We had already discussed that the cell has a distinct cell wall in case of a plant cell and many other cells. But if it's an animal cell, it will not have the cell wall. Then a cell membrane is typical to almost all cells. All of them have a cell membrane, also known as a plasma membrane. Then we talked about the next important organelle found in the cell, and that is the nucleus. Now, a nucleus, okay, will be well defined if we are talking about a eukaryotic cell. But if we are talking about a prokaryotic cell, the nucleus is said to be not well defined. Okay, so we'll come to this. What is the meaning of a not well defined nucleus? And you have studied that in your lower class, so we'll be revising that again. Now, just to tell you why is this nucleus so important to the cell. Okay, so a nucleus is important because it is supposed to be containing the chromosomes, which we all know are so famous because they contain the genetic material, which is the DNA. And in some organisms like the virus that's going around now, it is the RNA. Okay, so the nucleus is important because it has the blueprint of life. That means whatever the organism is going to look like or whatever functions it's going to perform is dictated by the genetic material, or is because the genetic material says so. Okay, so this genetic material is on chromosomes which are found in the nucleus. Cells could either be eukaryotic or prokaryotic, and we are going to look at the differences. Cells also have a semi-fluid structure which is called as the cytoplasm, which is filling the inside of the cell, inside or within the cell membrane. So cytoplasm is within the cell membrane, and it is said to be the main arena or the area of cellular activities. That means whatever chemical metabolic reactions are taking place in the cell are taking place in the cytoplasm. Now let's go to the next thing that you'd find in a cell, and these are membrane bound structures which are characteristic of eukaryotic cells. But when it comes to prokaryotic cells, as you all know and you pointed out to me in the first lesson, that they don't have membrane bound organelles. So this is the basic difference between eukaryotic and prokaryotic cell. This is one of the differences. So a typical cell should have membrane bound structures if it is a eukaryotic cell, but a prokaryotic cell, of course, you will not find these membrane bound organelles. And let's name some of these membrane bound organelles which you will find in a typical eukaryotic cell. These are the endoplasmic reticulum, the Golgi complex, the lysosomes, mitochondria, microbodies, and the vacuoles. Now, besides these membrane bound organelles, you also find non-membrane bound organelles such as the ribosomes. And ribosomes, as you might all be knowing and have already studied it in school, is that they are important because they are the site or the factories or the location where proteins are produced. So they are called as a site for protein synthesis. Now, ribosomes are not only found in the cytoplasm, but you also find it in organelles such as chloroplasts, mitochondria, and sometimes you also find them, chloroplasts, mitochondria, and you find them on the rough endoplasmic reticulum. Okay, so within chloroplast and mitochondria and on endoplasmic reticulum. Now, animal cells will also show other typical structures, like, as you can see there on my slide, the last point, centrosomes. We had already looked at the diagram and we had seen the centrosomes and the centrioles, and I had told you that they are important when it comes to cell division process. Okay, so these are few characteristics we have covered in this slide. Now, with respect to shape, and size of cells. Is there a similarity in all cells, or are there differences and variations seen in cells? So you all know that the answer is that cells differ. Some differ in sizes, some differ in shapes. So some examples have been given in your book. Mycoplasmas are smallest cells, and they have given you the size, the length of which is zero point 0.3 micrometers. And bacterial cells also are small cells but a little bigger than mycoplasmas, and they are around 3 to 5 micrometers. So these are some of the smallest cells found in the world. The largest cell in a multicellular organism is the single cell is that of the egg of an ostrich. So if you see the egg of an ostrich, it is a single cell and it's said to be a large cell. It's the largest isolated single cell. The red blood cells are about 7.0 micrometers in diameter, and all of you know that the red blood cells are found in the blood. They are also known as the erythrocytes. Another stat is given that nerve cells are the longest cells found in the body. So these are some examples to show you that the sizes vary. Now, even the shapes vary, as I told you in the beginning. You can have polygonal, you can have columnar, you can have cuboid if it is like a cube, columnar if it is column shaped, polygon, you know what it is, thread like if it is slender and elongated, or even it could be irregular. So there are different shapes of cells and there are different sizes of cells. Okay, so this was again supposed to be an activity, students, which you can do. Okay, so you will have to know what you will have to write down. Okay, the name that is hidden here, the name of the organelle that is hidden here, and wherever the red patches are seen, you have to identify these organelles. Okay, and write down in your notebook. So I will just give you about a minute to quickly identify and write it in your notebook. Okay, this again will be posted in classroom, so you will have to answer 5, 6 and 7, which is based on my earlier slides. Coming to prokaryotic cells, now we need to know the differences between prokaryotic and eukaryotic cells. So we are going to look at the structure of a prokaryotic cell in details. Okay, so prokaryotic cells, examples we studied in the earlier chapter: bacteria, blue-green algae, mycoplasma, and PPLOs, the full form of which is pleuropneumonia-like organisms. We also know that they are very small and they multiply more rapidly as compared to eukaryotic cells. We studied the shapes in the previous chapter of different bacteria. Bacillus is rod shaped, coccus spherical, vibrio is comma shaped, and spirillum is spiral shaped. Now, cell wall surrounding the cell membrane is present in all prokaryotes except for mycoplasmas, because we had also studied about mycoplasma characteristics in the earlier chapter and you knew that there is no cell wall here. Then there is a cytoplasm present, and we have discussed this point that there is a nuclear or nucleoid region, and there is no well-defined nucleus found in a prokaryotic cell. Now, the nucleus is containing the genetic material, which is the DNA, and in some prokaryotic cells, it could be also RNA. Now, many bacteria, in addition to this genomic DNA, they contain a DNA that is circular in shape, and I will show you this in the diagram. And this DNA is called as the plasmid DNA. Now, the importance of this plasmid is in engineering and biotechnology, which you will be studying in great detail in the 12th standard. Okay, so remember what this plasmid is. It is a small circular DNA which is found in many bacteria. Uh, the genomic DNA is found inside the nucleus, but the plasmid DNA is found outside the nucleus. It is found in the cytoplasm. So that's why I've listed here outside the genomic DNA. That means it's outside the nucleus in the cytoplasm, and it confers or it gives the bacterial cell a characteristic feature. It makes it resistant to antibiotic. So it is kind of protective to the bacterial cell, because if bacterial cells have the plasmid type of DNA, then they will be resistant to antibiotics. This has been seen in labs. Prokaryotes also have ribosomes. Their ribosomes are slightly different from that of eukaryotes. Then, prokaryotes also have a structure called as the mesosome. Now, mesosome is just an extension of the cell membrane inside the cytoplasm. And this could be in the form of vesicles, it could be in the form of tubules, or it could be in the form of plate like structures called as lamellae. So, we will look at mesosomes also in our diagram. So you have to remember that they're extensions of the membrane and they are infolded. That means inside the cytoplasm. Okay. So in our diagram now, you will have to look for the plasmid DNA. You will have to look for a nucleoid region, which is not a well-defined nucleus, and we will look for mesosomes. Okay, and organelles which are non-membrane bound, such as the ribosomes. So uh, before that, students, again a reminder to you, these will be listed in Google Classroom. You will have to answer them on your notebook. Yeah, before we go to cell envelope, this is a typical prokaryotic cell, and as I told you, we will look at the main structural details of a prokaryotic cell, which we have learnt in the previous slide. So there we had seen a nucleoid region, an absence of a nucleus, so you can see a no nuclear membrane, just the nuclear material is scattered in the cytoplasm in a region which is called as the nucleoid region. Then you can see the inclusions, and these inclusions, some are formed by the extended plasma membrane inward, and that is called as the mesosome. Then we also see the yes I talked about the nucleoid region, I talked about the inclusion, the mesosomes, the plasmid. Okay, so we said it is the DNA which is not here in the nucleoid region. In the nucleoid region, you will find the genomic DNA. It is outside in the cytoplasm. So this circular DNA found outside in the cytoplasm is called as the plasmid DNA. Okay, so also you can see inside the cytoplasm there are a lot of granules and inclusion bodies, and ribosomes, which are non-membrane bound structures. Now let's look at the outer covering of the cell of a prokaryotic organism. So going back to this slide, cell envelope. Cell envelope is the outer covering, whatever covers the cytoplasm of a prokaryotic cell, and it is said to have three layers. The outermost layer is the glycocalyx, the inner to it, that means the second layer after glycocalyx is the cell wall, and the layer inner to the cell wall is called as the plasma membrane. So just to go back to the diagram, you can see this is your outer glycocalyx. You can see the three layers in green. This is your glycocalyx layer, the white, which is the cell wall, and the innermost, which is in close contact with the cytoplasm, is the plasma membrane. Now this glycocalyx layer, coming back to it, could be a loose layer, okay, a very loose slimy layer. In that case, it's called as slime layer. So prokaryotic cells which have a very loose glycocalyx layer, it is called as the slime layer. Whereas some prokaryotic cells, the glycocalyx is very tough, like you can see in this diagram. Okay, in this image, it is a very tough glycocalyx, so we call it as the capsule. So please understand that the glycocalyx could be a loose layer called as the slime layer, or it could be a tough capsule. Now bacteria, dependent on whether this outer envelope, okay, that is your three layered cell envelope, gets stained by a stain called Gram stain, will fall into two categories. So if the bacteria is stained by the Gram stain, it is called as Gram positive bacteria. And if the cell envelope is not stained by the Gram stain, those bacteria are called as Gram negative bacteria. This is all you have to know about Gram staining. Okay, of course there's a lot of concepts related to Gram staining, but at your level they just want you to know the difference between Gram positive and Gram negative bacteria. Then you have, um, we talked about the glycocalyx outer layer. Let's talk a little about cell wall, which is inner to the glycocalyx. So this is responsible for giving shape and support to the cell, and it prevents the cell from bursting or collapsing. And finally, inner to the cell wall, as you can see in my last point, is the plasma membrane, and we use the term selectively permeable. Now selectively permeable means that this plasma membrane is able to selectively allow certain substances to pass through and keep certain substances out and not allow their passage. Okay, so this is the job or the function of the plasma membrane. So just to summarize here, three layers: outermost glycocalyx, inner middle cell wall, and innermost plasma membrane. Okay, so coming back to some of the characteristics, uh, features. Mesosome, I already explained to you, are the infolds of the plasma membrane, and in this diagram I showed you that inclusions could be either in the folds of the plasma membrane, could either be like this circular, a vesicle, or it could be like a plate, a lamellae, or it could be like a fine tube called as a tubule. This has to be kept in mind. So vesicles, tubules, and lamellae are all types of mesosome structures. Why is the mesosome important? Because it helps in cell wall formation and also DNA replication. The word replication means to make copies. Okay, when DNA makes copies of itself, it is called as replication. They also help in respiration, secretion processes, to increase the surface area of plasma membrane, and enzymatic content and reactions. Okay, now cyanobacteria is a type of prokaryotic cell which has chromatophores, and chromatophores are pigment containing structures. So therefore we know that the cyanobacteria also called as blue green algae because they have a pigment. And where exactly are the pigments stored? In this organelle called as the chromatophore. Next point, bacterial cells could either be motile or they could be non-motile. So if you look at this diagram, this is a motile bacterium. And how do we know it? Because it has this long elongated whip-like structure called as flagellum, and this is the locomotory organ which helps the bacteria to move from one place to another. Now you also have to know the three parts of the flagellum. So this part of the flagellum is called as the filament, then the small part is called as the hook, and the base which is embedded inside the cell is called as the basal body. So the three parts are filament, hook, and basal body. Some bacteria cells do not have flagella, they don't have cilia, they cannot move. They are called as the non-motile bacterial cells. Okay, now looking at the bacterial filament here, again this was supposed to be a revision for you. This is the filament, this is the hook, and this is the basal body. Now let's look at two other structures which you will also find in bacterial cell, fimbriae and the pilus. Okay, so going back to the diagram, you can see fimbriae are bristle shaped structures. Okay, and they are very important in attaching the prokaryotic cell to a substratum. So it could be a rock, it could be a stone. Fimbriae do the work mainly of attaching. And the pilus, which you can see, which is slightly longer than the fimbriae, this helps in bacterial sexual conjugation. Okay, so just remember the function of this pilus in conjugation. Fimbriae is for attachment. So that is what is listed in my slide here. Fimbriae helps in adhesion or connecting or joining to a surface, and pilus is meant for sexual reproduction, called as bacterial conjugation. Ribosomes and inclusion bodies in prokaryotes. Now, ribosomes in case of prokaryotes are of the 70S type. S stands for sedimentation coefficient. So it is made of two subunits. The 70S ribosome is made of two subunits, so you can break it apart. And the large part, one is a large part called as the 50S subunit, and a smaller part called as the 30S subunit. Then ribosomes are the site for protein synthesis, this we know. And at the time of formation of a protein, you will find several ribosomes attached to a single mRNA. Now this structure where a single mRNA is attached to several ribosomes is called as a polyribosomes or a polysome structure. It's like a chain mRNA with lots of beads of ribosomes attached to it. The next we need to know a little about inclusion bodies. So inclusion bodies are the reserve material stored in cytoplasm. So going back to this diagram, you can see here these are the inclusion bodies, and these could be storing gas, it could be storing food, it could be storing excretory products. So that is how the cell will store its reserved material. Okay, so in case of photosynthetic bacteria, you will find also pigment, and in case of certain blue green and purple green bacteria, you will also find gas vacuoles. So pigment are the cyanophycean granules. These cyanophycean refers to your blue green granules, which will give you color to that bacteria. Besides that, glycogen is a food product. Phosphate granules also are found. Inclusion bodies, also remember that they are not bound by any membrane and they freely lie in the cytoplasm. So this is end of part 2. We'll continue with part 3.